Good morning, everybody. How are you? I hope you're doing well with this situation that we live in. And today we are going to start Unit 8, which is all about the DNA and the relationship with the DNA and with the protein. Now, the first section we're going to study today is identifying DNA as a genetic material. Now, before I tell you the objectives, let me just explain this simple idea. Now, all of us, we studied genetic materials many times, and we know that genetic material is the molecule inside the cell that control all the cell's activity. Okay? Now, all of us, we know that DNA is the genetic material right now. But before that, people did not know what is the genetic material, whether the genetic material is a protein or DNA or carbohydrate. Nobody knows. Okay, nobody knew that. This is why our lesson, the beginning, is to identify DNA as a genetic material, to see what scientists did to reveal this amazing information that the genetic material is DNA. This is why our objective is okay. Now, the first thing that we're gonna study is we are going to describe what some scientists did, okay, to reveal this information about DNA and that DNA is genetic material. At the beginning, this is the first page of your book. I know some of you, he loses track or she loses track when she did not see a book page. This is why we are going to study first. We're going to study a Griffith findings. A scientist named Frederick Griffith, he did the first experiment that guided us to find the fact that the DNA is the uh, genetic material. Now, uh, what Griffith did? Let's study and see what Griffith did. Do you see this figure 1.1? If you want to look at it, take some notes on your page, just to know. Okay. Now, before I explain what a Griffith did, let me explain a very basic thing about a kind of bacteria that a Griffith used. Okay, Griffith, this scientist, he did an experiment, okay, to study and to find out what is the genetic material. Now, he used a bacteria called or caused a pneumonia. Okay. Now this bacteria has two forms. Okay. The first form is R bacteria. Okay. The second form is S bacteria. Now, if you compare both of those bacteria, you're gonna notice that the S bacteria has what? The S bacteria has a carbohydrate shell. Okay. A carbohydrate cover, comparing with the R bacteria that does not have this kind of cover. Now, what is the benefit of this cover for the bacteria? This cover helps bacteria to protect itself against the immune system of the host. Let's say this bacteria attack a mouse, okay? Now, if our bacteria attacked a mouse, okay, the mouse immune system will kill this bacteria, okay, because our bacteria does not have a shell, something to protect it, okay. But if S bacteria attacked a virus, attacked a mouse body, the immune system of the mouse will find a very difficult situation, will find difficulties to attack and kill this S bacteria. Usually, what happened that this S bacteria killed the virus. Okay? Killed the coronavirus. It's in my head. I can't take it out. But what happened is this S bacteria will kill the mouse. Okay? Take this in consideration when we explain Griffith experiment. Are bacteria weak? The immune system of the mouse can kill this bacteria and the mouse will not have pneumonia. S bacteria strong. The immune system of the mouse will not be able to kill this bacteria. The mouse will have pneumonia and the mouse will die. Now, the pneumonia is a disease that caused a problem, is a disease caused and affected the lung, okay, the respiratory system, okay. Now, let's see what Griffith did. Okay, now Griffith experimented with the bacteria that cause pneumonia. We said that. He used two forms of the bacteria, the S form, and we call it a deadly form, and the R form, which is not deadly. Okay. Now, a transforming material passed from dead S bacteria to lived R bacteria make them deadly. Now, we're going to explain this. Look here. I'm going to use my marker to explain some ideas. Now, we are studying the first case. Okay, this one that has circle around it. Now, what Griffith did, he brought a live bacteria, S bacteria, and he gave them to a mouse. What happened to this mouse? The mouse died. Okay, we're going to have, in this case, a dead mouse. Okay, that's the first case. The first step of his experiment, he gave a mouse, okay, he gave it what? He gave it S bacteria, alive S bacteria. He gave it alive S bacteria. What happened to the mouse? The mouse, okay, died. Now, the second thing he did is, which is the second one here, he gave another mouse, he gave it R bacteria, live R bacteria. What happened to the mouse? He lived. Okay? Nothing. He lived. He did a third step in his experiment, which is the third one. He killed the S bacteria, which means simply he collected the S bacteria and he boiled them. Okay, he heated them. In this way, he killed this S bacteria. Okay, what do you expect to happen? A killed S bacteria attacked a mouse. Okay, nothing would happen. The mouse lived. Nice, the mouse lived. Everything here makes sense. When you give a live S bacteria, the mouse will die. When you give a live R bacteria, the mouse will live. When you give heat-killed S bacteria, like in this case, the mouse lived because it's a dead bacteria. Nothing will happen to the mouse. Now, he did a third step, which is he had a heat-killed S bacteria. Okay, in the fourth step, heat-killed S bacteria. He mixed it with R bacteria. Okay, he had heat-killed S bacteria mixed with live R bacteria. Now, when they were separate, the heat killed alone did not kill the mouse. The R also did not kill what? The mouse. Now, when he mixed them together in the fourth one here, and he gave it to the mouse died. Now, he was thinking about this, and he said, there is something transformed or transported from the heat-killed S bacteria to the R bacteria that make that made this R bacteria able to kill the mouse. Okay, that's what he think about. And he called this thing a transforming principle. There's something went from the S, heat killed S bacteria, to the live R bacteria, and make this R bacteria to have what? To have a shell that protected. Okay, and he called this thing that changed this R bacteria, he called it what? He called it transforming principle. Now we all know that this transforming principle was DNA. Some DNA of the heat-killed S bacteria went to the R bacteria and changed the R bacteria to have what? To have a shell. And then it becomes stronger. We're able to kill the mouse. Okay. Now, I want you now to pause the video and to solve question number 1, okay, in the formative assessment, please. And then come back to me after you do this. Thank you. Okay, next time we are going to study the next experiment that did by a scientist called Very, okay, and he used the information from Griffith experiment and he modified the experiment, okay, to gain a stronger evidence about what is this transforming principle. What is this thing that went from the S virus to the R virus and make the R virus strong? Tamam? That's the point that we're going to study next time. Thank you very much, and we'll